For today's video, we're going to start genetics. And today, before we're going to start genes, alleles, genomes, and mutations, we're going to cover mitosis. Meiosis. We're going to differentiate what is mitosis and what is meiosis. There is eventually one main difference. Meiosis is about gametes. Okay, you should remember that it's all about gametes, while mitosis is about stoma. It's about somatic cells. Somatic cells are basically, for example, your hair, skin, or any damaged tissue. Where we wait a second. Where we should recover, for example, the lawyer, because when you have some injury, for example, on your elbow, we should maintain the surface of the skin in order to be able to be healthy, because if skin is open, it could lead to any injuries in advance. So mitosis is about somatic cells. It's something that you should just make a copy. While meiosis, this kind of stuff, is about gametes, egg cell and sperm cell. Why do we consider meiosis about egg and sperm cells? Because when we are creating a new offspring or new individual, we don't want two copies of baby A and baby A2. No, we want different species in order to have not species but offsprings, in order to have differentiation in evolution. You should reconsider the basic purpose of the life. So meiosis is about differentiation. It doesn't mean if you have one parent, parent A, it's mom, and we have parent B is dad, B is dad. We don't want to have the same baby A, and we don't want to have baby B, baby A, the second. We don't want that, because when you consider the evolution, it's more efficient to have a different diverse individuals, so the next generation could be different from this one or this one. So thus meiosis is about differentiation. So when we consider meiosis, we should think about meiosis. How I remember this. I see that mitosis also have this, but without T, we don't have T, and here is 2I and 2C. So I remember that there is two parts, prophase 1, prophase 2. Here is metaphase 1, we have metaphase 2. So this kind of stuff makes me remember that meiosis is about differentiation of gametes, and thus it has two steps. Okay, I think that it's just a hint to remember. And okay, let's start from prophase 1. Prophase 1 is about this kind of stuff. We see that prophase 1, like this is the cell eventually, and we have a nucleus with a DNA. Here we have a DNA. And in prophase 1, cell has 2 N chromosomes. So we had like 1X of DNA, like 100%, and we cannot divide it into 0.5 and 0.5X, like DNA. We cannot do that. Why? Because eventually and initially we had 1X, so here it should be also 1X. But how we are going to do that? It's easy, by just multiplying initial 1X into 2, so we have 2X. And here N is X, so we have 2N chromosomes. Okay. Just remember that. 2N, or in different words, we can say that it's double, like 2, double chromatid. Why chromatid? Because DNA is just one like spin or nitочка of something like just coded information, but this kind of stuff has the length of 3.5 meters, and it cannot be like transferred as this spin because it's too long. We have a very little cell where eventually even less space should continue DNA. So thus DNA is in form of chromosome, like just what we usually see in karyotype and other stuff. Basically it's more kind of this. Okay, and here we have a centromere. Well, not centromere, but locus or centromere. Yes, it's centromere. This is centromere. And the centromere appears as a constricted region of chromosome and plays a key role in helping the cell divide up in its DNA, like during the division. Because the centromere, remember we had the like spinners, so this centromere is the center of the spinner. So even so we will like rotated how many times, like many times, it will continue to have the center where you can handle the spinner. So this centromere also plays this kind of role because it's eventually the center. And prophase 1 does 2N of chromosomes. And this representation of chromosome is double chromatid, because when we say chromosome, we are considering that it's this kind of structure. So it does contain the DNA, and when we say locus of, for example, allele, we consider that this kind of part, on this one, this part of chromosome contains all alleles of, for example, skin color. Okay, And for example, this part of locus contains your information about the structure of hair. For example, just of hair. Like, if we will consider different chromosomes, this structure, and we will see a different, just let me draw it with a different color so you see that it's different chromosome. We have the same locus here and here. It's the part that we're looking for. For example, this chromosome is your, and this is your sister. And we see that this locus has alleles, for example, A and A. Okay, we see that dominant is, for example, white color, like white skin, and the little is for brownish, like brown skin color. And your sister, for example, has A and A, like recessive. To recessive, it means that it's eventually will be brown and brown. So her phenotype, like the physical appearance, will be brown color of skin. I don't know, maybe their parents of like the sister and your eventually parent has 2 A's and he is brown color. Maybe he's Indian or from somewhere else. And for example, your mom is European and has a dominant white skin color. So you will be like white, and your sister or someone else will be brownish. So that's how it works. But when we have a prophase 1, we should give, like, pass out this kind of information to the next generation. So it's kind of this. We had the square in genetics is the male. Male is, okay, it doesn't make sense. Male is square and female is circle. So when we see this kind of, when we see square plus a circle, it means that it's couple, like it's couple, married couple, whatever, we don't care. And then we see that they have their own offsprings, like kids. And we see, for example, there is, in their family, it is boy and girl, or maybe 2 boys and 1 girl. And in order to be able to differentiate them, in order to be able to determine whether it's your kid or not, like you are going to pick up your kid from kindergarten, and you should be able to differentiate which of this kind of many kids, which one of them is yours. So you should be able to see some similarities to you or to your wife, for example, if it's the male who picks up the kid. So prophase 1 makes this person and this person gametes, like information that they are going to pass to N. So they can have the next gametes and have genetic information which they are going to pass furthermore, and the result will be like this kid. But we cannot say like once they passed their genes for one kid, it's over. No, they cannot have another child. No, we can say that it's possible to have another child. So thus they have to have one N of DNA. And also pass N here. So they can also have a partner and like have their own offsprings. Okay. So prophase 1 makes 2N chromosomes. And this kind of, if we say that this is chromosome, 2N chromosomes or double chromatids is about making this paired like this. And we see that this and this chromatid, one is chromatid now, chromatid, is homologous, means they are the same. They're the same, so it's just a copy, okay? And we say that in prophase 1 there is crossing over occurs. Why? Because when we have different kids, like one family can have 3 kids and none of them are going to be the same. So eventually your brother and your sister and you are going to be different. Like he has brown straight hairs, he has like brown and some kind of curly hairs, curly, while like your sister has a little bit like dark hair and she has like totally curly hair. So we can say that probably one of the parents are brown color hair and has a straight hair, and maybe he has like dominant, for example, if we say it's straight, no, it's basically, it would be much better to write different, like to pick up the letter of alphabet that is different for being capital and the small, because when you write like C and C, you will be messed up. So when we are writing dominant and recessive, we are going to choose this kind of couple literals, like D and D, for example. Which kind of R usually is used? Yes, it's commonly used, this kind of pairs. So, for example, this kind of man has straight hairs and has inherited a recessive gene that is about being and having a curly hair. And for example, this kind of woman, has also A and A. So it means that he has a dominant allele of straight hairs. If he has curly, that it means that he has a recessive, and she also have recessive. That kind of, so this is the way how it works. It's pretty easy, basically, when you understand the logic behind this. So in order to have a different kind of hairs in every kid, we're going to have a crossing over. Crossing over, it means that, for example, if this copy is red color, for example, it would be easier to remember, and this is white color, like original is white, yellow, and this is a copy, like replication. We can say that crossing over is about giving up some information, so exchange of information. So the next, like this kid had inherited like this kind of copy with a little of information of original. Okay, and this kid had this chromosome that is inherited. So thus he has a straight and he has a curly. Okay, so crossing over is about making up this differentiation in the next offspring. So thus it's very important. Okay, we covered prophase 1. It's about 2N, like double chromatid. If we had one chromosome, then we had 2 chromatids. Then we have a homologous. It means that they are the same, like they have the same structure, they have the same pattern of genes, and like they have the same location of genes, locus. It means that the pattern of gene is like A, G, U, G, C, and the copy is like this kind of pattern of genes had this chromatid. It means that this chromatid also going to have A, G, U, G, C, okay? That means homologous. And locus, it's about like if here of chromatid, here we have genes located for color of eye color, so it means that this chromatid also going to have the locus like right there. It's also going to be about the eye color. Okay, we're going to move on metaphase 1. It's all about, you see, the cell is preparing to divide. Okay, so it's easier to divide when let's pretend that you have a lot of kids and you are responsible for them. Okay, like this is the room and you are there, you're responsible, but you have many kids and they're going to shout, they're going to run, they're going to be messy, they're going to be they're going to scream, cry, and all that stuff. So it's easier to you to not look around and make this kind of strict decisions, but it's easier for you to have them line up, so you can have some kind of control over them. So metaphase 1 is about this kind of control. Okay, we don't want to have this kind of chromatids, just they're all about nucleus. You should look up from them, you should just find out where is it. No, why do we do that when we have an option to make them line up on the equator? Like if it is like it's not, if we have the cell and the cell have equator, like the center, just as Earth have the equator, so the cell also have equator. So in metaphase 1, this 2N of chromosomes are going to line up on the equator. Okay, and as they're going to line up on the equator of the cell, the spin, like this dot, yes, that I draw, is this kind of structure. Okay, just remember that is the same, just we are looking far apart. So as they're lined up on the equator, and we have to divide them into two cells which have own part of DNA, we have to divide them. Okay, but how we're going to divide them? We're going to divide them by spindle microtubes. Spindle microtubes are going to be like originated from different like the north and the south, like north and south parts of our cell, and they're going to come up with this kind of microtubes for every pair of our double chromatid. Again, this also okay, let's draw it again. Okay, this and this north will have this one, and as I said, it is about this kind of structure. Yes, so one is going to be pulled apart by one microtube, and another going to be pulled by another microtube. So they can divide in one cell and another cell. Okay, like they move homologous pair to equator of a cell, and orientation of pattern and material chromosomes on either side of equator is random and independent of homologous pair. So it means that each of the pair is going to be divided. It's eventually this. Okay, let's move on to the anaphase 1. So as we lined up all our genes into the equator, and we prepared spindle microtubes for each of the pair, the anaphase is eventually going to divide them into two different poles, like the south and the north. So this homologous pair, which was in this kind of structure, is going to be just pulled apart. Okay, they're going to be apart from each other. So when you will see any tasks, it would be looked like this. So anaphase is about being separated. Metaphase is about being on the equator, and prophase 1 is about making double chromatid. Okay, double number of chromatids. And we're going to move on telophase 1, and eventually we are going to finish with like the first part of meiosis. Telophase 1 is about, we have a two okay, no, we have we should make it bigger. We have one, it's yet is one cell, but the spindle tubes already made the pair of chromatids separated. So if it's one, there is one. Okay, there is one, there is also one, there is also one. This kind of stuff is repeated many times. So it's anaphase, like anaphase. But telophase is about chromosomes uncoil, During interphase that follows, no replication occurs, so there is no more replication. We are working with that we had to work with, and reduction of chromosomes number from diploid to haploid completed. So if we had 2n chromosomes, in order to make 2 cells with one pair of n, 1n, we completed this part, okay. And cytokinesis occurs. What is cytokinesis? It's about creation of this kind of new cell membrane, cell wall, no membrane, so they can divide, because when you have like the pool and you want to dry out this one but not this one, you're going to build the wall, so not even the millimeter of water can flow from this to here or from here to there, so they can be dried. And here's also the kind of situation where we can divide the cell. So telophase 1 is completed. The second part of meiosis is about to repeat all the parts: prophase, metaphase, anaphase, and telophase. Okay, it's going to be second, and also second. Prophase the second, okay, or prophase 2, is about chromosomes, which still consist of 2 chromatids condense and become visible. So the main difference, I am sure and 100% sure that you will have this kind of task in the summative and informative, and also in the classwork, and also in IB exam. You have to be able to differentiate the phases of meiosis, like prophase 1, prophase 2, based on just this kind of visual. But how we're going to make this is very, very easy, okay? Prophase 1 is different from prophase 2 by the fact that here we see more tied up dots, these dots will be more bold and visible for you in prophase 1, and there is not any division, so you do not have this kind of division. You just have the cell with visible bold dots of chromosomes, so it means that it's 2n. But in prophase 2, there is already cytokinesis occurred in telophase 1. So prophase 2 has this kind of membrane between the cell, like in the cell. And the dots are bold, okay? They are bold and they're together. So we see that it's prophase 1. Prophase 1, we draw this kind of cell, has chromosomes, like this part has chromosomes. These dots are chromosomes. It's chromosomes that still consist of chromatids, okay? It still has chromatid. So if this was pulled apart and this was pulled apart, now we have just this kind of structure. but we should also divide this into different parts. So they're going to be pulled apart. And prophase 1 also prepares this structure, chromatid, to be divided. So as it is going to be divided, it should be more visible. But the main difference between prophase 1 and metaphase, or prophase 2 and metaphase 2, which can be kind of scary to make. Prophase 2, it would be more like that, while in metaphase 2 it's going to be kind of this structure. Okay, you see. So also metaphase 1, metaphase 2 is about lining up on the equator and division by the spindle tubes. So we see here is this kind of cytokinesis occurs where, and we have a cell, and they are going to be pulled apart by spindle microtubes. So it's about metaphase 2. Anaphase 2 is about separation, and the structure that we had, chromatid, is going to be finally pulled into different poles. So we have now, we had one cell, okay, we had one cell which divided in the end of telophase 1, which divided and has the structure of this kind of structure. Oh my god. This kind of structure. I will not draw many of them, just 1, 2 different sides. And anaphase 2 divides, okay, I should draw them like this. They're going to be here and here. So anaphase 2 makes them separated. So here we have just a tiny part. So here we have also the tiny part. Anaphase 2 is dividing them into different poles. So it's south, south, north, north. While the telophase 2 is also about creating cytokinesis. So we are forming a new wall between them. So it's eventually 2 different walls. And as we are finished with dividing the structure of double M chromatids, we're going to form again a nucleus. Okay, the DNA should be a nucleus. So here in telophase 2 is going to be formed a nucleus. It is like the final step. Okay, so eventually this is all. And as we had crossing over in prophase 1, like in the very beginning of this has, okay, it is this kind of wait a second, just so as we divided this two chromatids which had crossing over. So thus this cell, like the cell 1, cell 2, cell 3, and cell 4, is going to give a different kind of DNA to each of new offspring. Okay, so by this steps, all of the steps, the kids that is born in one family and have one couple of parents, like the same parents for every kid, they are going to be different, okay? Because they had differentiation in prophase 1, and this leads to the telophase 2, which have formed 4 new cells, haploid cells, haploid because they are going to have 23 chromosomes, and they are going to have another pair of 23 from your mom and dad, so you can have a full of 46. Okay, so this have 23, this have 23, and let's say that it was formed in female, thus it's meiosis of egg cell. Okay, and let's say that this 23 pair is this pair. That's all.